Hello everyone, in this video we will talk about beta-oxidation. In this process, the goal is to bring free fatty acids to the tissues that need it and break it down in the mitochondrial matrix to make acetyl-CoA. So now what tissues actually use free fatty acids for energy? Well, the answer to that question is the heart, the skeletal muscles, and the liver. The liver can use the acetyl-CoA it makes from beta-oxidation to make ketone bodies. So here is a diagram of a cell with the cytoplasm and the mitochondria. The mitochondria has an outer membrane, an inner membrane, an intermembrane space between the two membranes, and the mitochondrial matrix in the middle. The mitochondrial matrix is where the breakdown of the fatty acyl-CoA will take place, which we will see later in this video. The free fatty acid, the long-chain fatty acid that we made from fatty acid synthesis, is the 16-carbon saturated palmitic acid. We can make longer ones and even unsaturated fatty acids, but let's take a look at the 16-carbon saturated fatty acid that we already saw how to make in the previous video. This fatty acid has to get into the cytoplasm and eventually to the mitochondria of the cell to undergo beta-oxidation. Remember, a fatty acid has an acid end with COO-, right? That's the carboxyl end. So it's a negatively charged molecule. And it also has an alpha carbon and a beta carbon. The alpha carbon is next to the carboxyl carbon, right next to it, whereas the beta carbon is 2 carbons away from the carboxyl carbon of the fatty acid. The goal in this process is to convert the fatty acid into an acetyl-CoA and a bunch of ATP. So the palmitic acid is 16 carbons, but since I already drew 3 carbons here, the R group has an additional 13 carbons. So in total we have a 16 carbon chain. So let's blow up the cell and let's look at what happens right over here. We have the cytoplasm over here, and we have the mitochondria right over here. We have the outer mitochondrial membrane, the intermembrane space, the inner mitochondrial membrane, and the mitochondrial matrix. Here is the fatty acid with the negatively charged carboxyl end. It already got inside of the cell. So the first thing that we want to do is activate that fatty acid. Now the question is, how can we do that? Well, in order to activate that fatty acid, we have to turn it into a fatty acyl-CoA. And the enzyme that will help us do that is fatty acyl-CoA synthetase. This is a crucial enzyme that will help with fatty acid activation. In order to activate our fatty acid, we need to first add the COASH or the coenzyme A to it, as well as an ATP molecule. The ATP will actually be broken down to AMP and pyrophosphate, which will separate into 2 inorganic phosphates. This is because the negative charges of the phosphate repel. So once you have the pyrophosphate, which is 2 phosphates stuck to each other, they're gonna just split apart. What this all means is that actually this requires the equivalent of 2 ATP for the activation. So the ATP goes directly to AMP in pyrophosphate, and then the pyrophosphate splits to 2 inorganic phosphate. This is the equivalent of 2 ATP molecules. And here is the fatty acyl-CoA, which we will be producing from this activation step. So instead of having the O minus, you have the coenzyme A group attached to it. So now we have a fatty acyl-CoA. So this enzyme, fatty acyl-CoA synthetase, turned the fatty acid into a fatty acyl-CoA. Adding the coenzyme A actually traps it within the cell so it cannot leave. This is super important as now we can use it once it's trapped. There is a protein on the outer mitochondrial membrane actually sticking to the cytoplasm known as CAT1 or carnitine acyl transferase 1. It can also be called carnitine palmitoyl transferase 1. If you recall in the fatty acid synthesis video, malonyl-CoA actually inhibits this protein. This is actually the enzyme that will donate a carnitine to the fatty acyl and remove the COASH group. So this is a very, very important molecule. So you end up forming this fatty acyl carnitine, which can enter the mitochondrial membrane through a translocase protein. After doing some reading, I realized this translocase is on the inner mitochondrial membrane, as the outer membrane is porous, so the fatty acyl carnitine doesn't need help going through it. But the inner mitochondrial membrane is not as porous, so there is a translocase so that the fatty acyl carnitine can get in. Next, there is another enzyme, CAT2, carnitine acyl transferase 2, or carnitine palmitoyl transferase 2. This is also inhibited by malonyl-CoA. CAT2 will resupply the COASH and cleave off the carnitine section, which will then be recycled. It will go through that same translocase on the inner mitochondrial membrane and be recycled into the cytoplasm. So now we actually get the fatty acyl-CoA in the cytoplasm. So let's look at this process again. Here we have our cytoplasm, and inside we have the mitochondrial matrix. We start off with a fatty acid that has entered into the cell and now it's in the cytoplasm. Then COASH will be added and ATP is required to activate it into the fatty acyl-CoA. And this requires an equivalent of 2 ATP. And the activation requires the enzyme fatty acyl-CoA synthetase. The CAT1 on the outer mitochondrial membrane will add carnitine and take away coenzyme A to liberate the fatty acyl carnitine, which will make its way into the mitochondria with the help of the translocase on the inner mitochondrial membrane. The CAT2 will also be on the inner mitochondrial membrane closer to the matrix, and it will give back the COASH, the coenzyme A, and take away that carnitine, which will be recycled as it makes its way through the translocase and into the cytoplasm of the cell. The resulting molecule that will be formed is actually our fatty acyl-CoA, which I put in yellow right over here. This fatty acyl-CoA is now in the cytoplasm and is ready to undergo a process of beta-oxidation. The fatty acyl-CoA will undergo beta-oxidation and each cycle make an acetyl-CoA, except in the last cycle where you make an extra one. In the last cycle, you actually make 2 acetyl-CoA instead of just 1. Every cycle, you're also going to make a fatty acyl-CoA, which is 2 carbons shorter than the previous one. And you're going to make NADH and FADH2. And of course, from these, we can get a bunch of ATP, which we'll look at later. So the acetyl-CoA that's produced can then enter the citric acid cycle or the Krebs cycle to make NADH, FADH2, and ATP. And we will look at that later. You actually make 3 NADH, 1 FADH2, and 1 ATP via substrate level phosphorylation via the citric acid cycle from 1 acetyl-CoA molecule. So this is a very, very useful process to really generate a lot of ATP. So now let's just go up right over here and take a look at what happens in the process of beta oxidation. So in beta oxidation, we are actually going to start with our fatty acyl-CoA, which is present in the cytoplasm, and it's ready to undergo this process. The first step is carried out by the enzyme acyl-CoA dehydrogenase, which uses FAD and turn into FADH2. In this step, we actually form a double bond between the alpha and the beta carbon. Remember, we had the alpha carbon next to that C double bond O carbon, and the beta carbon, which was 2 carbons away. So you form a double bond between the alpha and the beta carbon. This is an oxidation step because the fatty acyl-CoA gets oxidized, whereas FAD gets reduced to FADH2. So in this step, the FAD takes the H's from the fatty acyl-CoA and puts them into FADH2. So the resulting molecule has 2 fewer hydrogens. Now, what is that molecule called that we actually form? This molecule is called trans delta 2 enoyl CoA. So this is the molecule that we are going to form right over here. This product will get hydrated with water using the enzyme enoyl CoA hydratase. So hydratase is an enzyme that adds water, and in this case across the double bond. So in this case we will form our beta hydroxy acyl CoA. And the beta hydroxy acyl CoA has the hydroxyl group on the beta carbon. In the next step, what will happen is this beta hydroxy acyl CoA will turn into a beta keto acyl CoA with the enzyme beta keto acyl CoA dehydrogenase. In this step, the H and the OH in blue will be lost and donated to an NAD+ to form NADH. As a result, we convert the beta hydroxy group to the beta keto group. And this is our second oxidation step, where we bring those 2 hydrogens, we put them onto NAD to form NADH. So we oxidize this beta hydroxy acyl CoA into the beta keto acyl CoA. We lose an H here and we lose an H on the hydroxyl group. So we turn it into a ketone group right over here on the beta carbon. So again, we began off with the acyl-CoA dehydrogenase, the first oxidation step, giving us FADH2, putting that double bond between the alpha and beta carbon. Then we add a water across the double bond to form our beta hydroxy acyl CoA, because we have a hydroxyl group on the beta carbon as we added the H and the OH, the water, across the double bond. In the next step, we actually lose 2 H's. We lose 1 H here and 1 H from the OH, and we form a beta keto group now on the beta carbon. And for that, we use another enzyme, another dehydrogenase, and we make NADH. Now, in the last step, we use a Thiolase enzyme to add a coenzyme A in the form of COASH and cleave off the fatty acid between the alpha and beta carbon to give us the fatty acyl CoA that's actually 2 carbons shorter. So we cut right over here, and now we give it a coenzyme A, so we form this fatty acyl CoA that's 2 carbons shorter, minus 2 carbons. We also form an acetyl CoA. Think of it this way: we add an H to this carbon, so now we have CH3 C double bond O coenzyme A, and this molecule is known as acetyl CoA. So in this process, using the Thiolase enzyme, we give it the coenzyme A and we form a new fatty acyl CoA, which is 2 carbons shorter, and the acetyl CoA. Now, coenzyme A was added as we had 2 coenzyme A groups in our product, but only 1 in the reactant, beta keto acyl CoA. So the Thiolase enzyme had to donate another COASH group. So here's a question: how many acetyl-CoA will be made from a 16-carbon saturated fatty acid. So we will chop it 2 carbons at a time. And to calculate the number of acetyl-CoA that you make, you simply divide the total number of carbons by 2. So 16 over 2 is 8. However, we make 7 cuts, so only 7 rounds of beta-oxidation. And we make NADH and FADH2 per round. So we make 7 NADH and 7 FADH2. The formula is the number of acetyl-CoA minus 1. So 8 minus 1 is equal to 7. And remember, in the last round, we make an extra acetyl-CoA molecule. So we make an extra acetyl-CoA. So 7 rounds, but we make 8 acetyl-CoA. So what is our total energy yield that we will make? How many ATPs will be formed? Well, we start with palmitic acid, which is a 16-carbon saturated fatty acid. And remember, we activated it to form fatty acyl-CoA, which costed us the equivalent of 2 ATP. So we have to take that into account. The acetyl-CoA gives off 3 NADH, 1 FADH2, and 1 ATP in the citric acid cycle, or the Krebs cycle. Since we make 8 acetyl-CoA, the total NADH that will be formed is 3 times 8, which is equal to 24. The total FADH2 is 8 times 1, which equals 8. And the total ATP is 8 times 1, which equals 8. In addition, we make 7 NADH and 7 FADH2 per 7 rounds of beta-oxidation, because each round we make 1. So from 7 rounds, we make 7 of each. 7 NADH and 7 FADH2. So in total, we make 24 plus 7, or 31 NADH, 8 plus 7, which is 15 FADH2, and 8 ATP. But we use 2 ATP for activation, so we have to subtract it. Since we get 3 ATP per NADH and 2 ATP per FADH2 in the electron transfer chain, the total is 31 times 3 plus 15 times 2 plus 8 minus 2 for a grand total of 129 ATP molecules. So that's a lot of ATP from fatty acids. Now, the interesting thing is there's actually an acronym to help you remember the beta-oxidation process. And that acronym is OHOT. Now, the O stands for oxidation. The H stands for hydration. The other O is oxidation again, and the T is the thiolase. So the first O is where we get our FADH2 from the acyl-CoA dehydrogenase. The H is the hydration, where we hydrate that enoyl-CoA to form the beta-hydroxy group, right? The beta-hydroxy-CoA. And then is another oxidation step, another O, where we make our NADH. That's where we turn the beta-hydroxy to the beta-ketone group. And then is the thiolase, where we add the coenzyme A, and that's where we actually cleave our fatty acyl-CoA to make another fatty acyl-CoA that's now 2 carbons shorter, and we give off an acetyl-CoA. So OHOT is a nice mnemonic to remember all the steps of beta-oxidation. The last thing is to realize what happens if we have unsaturated fatty acids. Well, if we have unsaturated fatty acids, that means we have double bonds. We actually end up making 1 less FADH2 per double bond. So you actually end up making 2 ATP less per double bond. So a saturated fatty acid gives us more ATP than an unsaturated fatty acid, assuming you have the same number of carbon atoms to work with. Now, what happens if you have odd chains? That's the other question. Well, if you have odd chains, at the end, during the last round, you end up with propionyl-CoA instead of acetyl-CoA. So if you have an odd chain, at the last step, you're going to make propionyl-CoA. So let's say you have a 17 carbon molecule. So you know that for a 16 carbon molecule, you make 8 acetyl-CoA molecules. But for a 17 carbon molecule, you're actually still going to have 7 cuts. But in this case, you're going to make 7 acetyl-CoA, but the last one will not be an acetyl-CoA, it will be 1 propionyl-CoA. So that's the only thing I wanted to point out. And the other interesting thing is that this guy here actually gives us less ATP than an acetyl-CoA. And we will look at that in the next video. In the next video, we'll also look at specific beta-oxidation that happens in the peroxisomes. So stay tuned and hope you found this video helpful.